Hi, my name is Yana Bromberg, and I'm going to talk to you today about the effect that deep learning can have on figuring out the metagenome functionality. So first things first, what's a metagenome? Well, we know microbes are everywhere, and they exist in these communities that happen to live on the skin, in the gut, maybe even in water samples or in soil of the particular environment. And since we can't really apply available microbial analysis techniques in order to try to figure out what are these microbes and particularly what they're doing in the environment that they're in, one of the things that we've been doing is simply collecting their genomic data as a signature of a particular environmental niche. So let's say the human gut or a water column. We then sequence the DNA that's found in this sample, and then we can start to do some analysis. One of the easiest types of analysis that we can do is trying to reconstruct who is there. Now, I say easiest because that's the easiest to understand, but it's super complicated to do that. So if you imagine shotgun sequencing, so breaking down this collection of microbial genomes into these reads, and then trying to assemble who was there in the first place, you will see that this is a very difficult task. In fact, the genomes that you would most likely be able to assemble are the ones of the bacteria that are most present in a particular community. Now, this is a very lossy situation, which means that there are plenty of organisms that you would like to be able to sequence, but you never get the chance to because there is very few of them. So another way to think about how we're going to annotate metagenomic data is via alignments to known organisms. So you can imagine a set of bugs that have, let's say, the red and the blue one here that have reference genes and so corresponding reference proteins. And each one of these genes or proteins has a function assigned to it. So the blue organism, for instance, has two functions, F1 and F2, and the red organisms has two functions, F1 and F3. So here it's important to recognize that two different organisms can carry out the same activity, and that's the F1 in this case. Now, if we have this collection of reference genomes, you can imagine that we can sequence reads from a metagenomic sample. And we know that these reads come from genes or proteins that would have been encoded in the corresponding organisms. It could be one organisms or 10, it doesn't matter in this case. So how would we figure out which genes would have been there? So one way to do this is to say, okay, this read can align to a gene that does F3. Therefore, the gene that actually had given rise to this read is also doing function F3. Here, F1 could align to this read, and it actually wouldn't matter which organism this particular read aligns to, because in the end, all we care about is the fact that this is going to be a gene that carries out function F1. Now, you're going to need a very particular type of analysis to do this sort of alignment-based transfer. And we had actually done this. We built a tool called mi-faser, which I'm going to briefly mention shortly. Now, of course, the problem here is that you can't annotate anything that you don't have references for. And also there is going to be a situation where the read doesn't actually overlap in its completeness with the gene that is being referenced. This last problem is going to be highlighting the situation where the reads from homologous sequences, which could potentially be annotated, are not always going to be sequence similar. So for instance, if you look here, we have the tetracycline repressor protein that comes from C. aceticum, and here one that comes from E. coli. Now, if I align the two sequences, the aligned region is just this blue line over here. So of course, if my reference protein is this orange one, and then I query with the read that comes from the green protein or the green gene, what I'm going to see is that if that read comes from this region, it will actually do fairly well in recognizing of the orange functionality. However, if it comes from this region, it will not. Now, so this is, as I said, one problem. The other problem is the lack of annotation. And if we consider the SwissProt database, which is our biggest repository of manually curated sequences, what you will see is that out of the 550,000 proteins that have been manually curated, not a very large number if you consider in light of all other available sequences. But even for this set, only 335,000 come from bacteria, and even fewer still, so only 22,000 come from annotations of bacterial proteins, so evidence that this protein actually exists. Of these 22,000, 13,000 happen to have an enzymatic activity annotation in the form of the enzyme commission number, which I'm going to come back to slightly later. But this annotation is predicted in many cases. In fact, if I asked for experimentally annotated EC number annotations, we would see that only about 7,000 sequences carrying 1,800 or nearly 1,900 diverse functional activities can be annotated. So that means that if I was looking for reads that actually map to these sequences, unequivocally map to these sequences, in order to try to figure out which EC numbers, that is functionalities of the genes, exist in the metagenome that I'm looking at, I would only be able to annotate about 2% of an average metagenome that I've collected. So I was promised you to tell you a little bit about this tool that we built. So Chensheng Zhu was a student and a postdoc in my lab, was the guy who had created mi-faser, which is the method that annotates functionality of the reads, granted for only 2% of the metagenomic data, but it's very accurate. And what you could see here is that actually for the vast majority of the ranges for the faser functionality, it outperforms just basic BLAST. And so we use this method quite extensively in order to compare the functional abilities of the individual metagenomes. But of course, 2% is not 100%, and we wanted to see if we could annotate more metagenomic data using more advanced techniques. So another postdoc in my lab, Adrian Horfrost, had decided to come up with a model that models the language of prokaryotic life. She trained an LSTM RNN called LookingGlass to try to recognize the next nucleotide in a particular genome sequence so that she can embed the genome world into the bacterial genome world into a higher dimensional space. So here you try to predict the next nucleotide, right? So the same thing that many people do in natural language processing. And what we found is that LookingGlass is actually better trained. So the x-axis here is the epochs and the y-axis is the accuracy. So we never get above 40% accuracy in predicting the next nucleotide. But what we noticed is by selecting a data set that is fairly diverse, that is a single organism out of one class of microorganisms. So class is the second level of the phylogenetic taxonomy here. So having this diverse collection of microorganisms actually gave us a fairly good model, or as good a model as we were going to get in a much shorter time than increasing the set size to even, let's say, 10 million in dark blue over here, or 18 million that's in green here. So we were successful to a certain extent in embedding the reads into this space, into this language space. So then we wanted to ask, what is it that the model captures? And we asked this in a way where we said, okay, how are the embeddings of the individual reads going to differ? And what we found is a very curious thing, right? So if we take the distance between any two given embeddings, we see that if the reads come from homologous sequences, then the embeddings are actually closer together. So the embedding similarity is the reverse of the embedding distance. So this blue thing here indicates that embedding similarity is higher for homologous sequences than it is for non-homologous sequences in red. However, we found that this information is actually orthogonal to sequence similarity. So what you see here is the distribution of the homologous in blue and the non-homologous in red sequences across the embedding similarity space on the x-axis. While here on the y-axis, as you're seeing sequence similarity, again, homologous in blue, so higher sequence similarity suggests more homologous sequences. And you see that these distributions are quite different. So basically, if we wanted to really go down in the amount of sequence similarity that we were going to actually allow for calling something homologous or not, we would need to take advantage of the embedding thresholds and basically cut off this piece of information that's sitting on the left side of the embedding similarity threshold. Now, we would still have some errors, but remember, this is at a really low sequence similarity. So we could use embedding similarity to drive our homology recognition. However, you see from this example that the embedding similarity doesn't necessarily capture sequence similarity. So what does it capture? And one thing that we found is that if we looked at the individual environmental packages or environmental niches, such as, let's say, the microbial mat or biofilm in pink over here, or the human gut in purple, versus the water over here in light green, or plant-associated microbiomes in green, in dark green, we see that the reads that belong to a particular package tend to have a closer embedding distance than the ones that belong to other places. So basically, reads that come from the water are going to group with other reads that come from the water microbes, whereas reads that come from the human gut over here are going to look like other reads that come from the human gut. So this is simply looking at the embedding distance of the individual microbiomes. We were also able to train a downstream task of predicting the first, second, and third EC digit as identified by mi-faser using the embeddings. So basically, we trained a downstream task, a downstream model on the embeddings of the reads that have been annotated with mi-faser labels. And we saw that we are able to make predictions of the first EC digit, the second EC digit, and the third EC digit. So sequentially more refined functionality by simply training these models. So without any alignments, by predicting mi-faser annotations. Now, mi-faser is not 100% accurate. So we wanted to see if we could reflect on the actual activity of the individual metagenomes using tools that are not predictive by themselves. So we trained a model using embeddings again, in order to predict whether there is this oxidoreductase activity encoded by EC number 1, right, that is present in a particular metagenome. Now, we don't have the gold standard for these metagenomes, because if we did have the gold standard, then we wouldn't be making any discoveries. So what we wanted to see if we can use our predictor to try to identify new EC1 activities which weren't seen before. And we took a couple of metagenome samples, and I'm going to talk about them in a second because that's going to be important here. And we annotated them using mi-faser, using mgrest, and using this predictor that we trained to call the reads as being oxidoreductase or not. Now you can see with mi-faser, we're only getting about 2% of the annotation. We have a lot more with mgrest, and all of the reads can be annotated as oxidoreductase or not by the EC1 predictor. Now there's a lot more EC1s here, and this is actually a question of how good is this prediction? And then if we go back to where this data came from, we see that for the surface activities of the metagenome versus the oxygen minimum activities of the metagenome, there should be a difference because there is different functionality of the microbiomes in all of these different spots. So at the surface of the ocean, at the oxygen minimum zone versus the mesopelagic region. But we don't actually see any difference for either mi-faser or mgrest. Now, in reality, you would expect that there would be a lot more oxidoreductase activity in the oxygen minimum zone, because there you would actually need to do more work to get those electrons out. And this is something that we observe when we use the EC classifier trained on the LookingGlass embeddings. So this we use as a confirmation of the fact that EC1s predicted by the LookingGlass predictor are actually capturing the functional signal expected for this microbiome community. So some conclusions: metagenomic functional differences across samples are important. And embedding distances are orthogonal to function transfer by homology. So basically, we have this new way of understanding the functionality that we know, even though this is not discovering new functionality, this is discovering new proteins that can carry out the functionality that we already know. Okay, thank you. This is the people that did all the work. Adrian and Chencheng I already mentioned, as the rest of the lab. And we hope to see you in the lab too. So we're currently hiring. Please let me know if you're interested.